You're all very welcome. Thank you for coming to hear about what we've learned as we have pursued our investigation and development of products around liquid biopsy. We are very interested at QIAGEN in liquid biopsy because we believe it has the power to revolutionize healthcare because it offers so many benefits to the patient, to the pathologist, the oncologist, and actually to researchers. And we'd like to share what we have learned, why we think it is so important, and then I will hand it over for some real-world examples from Dr. Laurence Lapointe of Clinical Genomics, who has worked with us as we have proceeded with this journey. So what is liquid biopsy? And for us, the definition that we use is it's a minimally invasive technique to help detect molecular biomarkers from blood, urine, or other bodily fluids, which allow for improved diagnostics and treatment of cancer and other diseases. And there's really three main forms that you would look at. The free circulating nucleic acids that may come from dead cells that are shed into the bloodstream, and you can track cancer-related mutations, or exosomes, which are the tiny microvesicles found in body fluids that transport RNA between the cells, and then circulating tumor cells that may be indicative of cells being shed into the bloodstream, and those cells will carry the genetic information. So it may be something where you have a recurrence and you're seeing new tumor cells being shed. And these sample types are much more accessible to and less invasive to the patient compared with doing an actual biopsy using fresh or frozen tissue. And so why we believe it has the potential to transform healthcare and is because it holds so many benefits for the patient. For a patient, it's safer and less painful, and it allows you to monitor more frequently. You can monitor as frequently as you want. For the pathologist, you get faster patient results, and it allows easy monitoring because you will have easy access to the samples. And you can then track as you're developing you can track if there is drug resistance coming out or a tumor recurrence. And then for researchers, there will be more samples available, and you can go back to some of the stored samples, so allowing retrospective studies on the existing samples, and hopefully identify disease-related biomarkers in a simple, non-invasive way for the patient. And what QIAGEN has tried to develop is reliable, best-in-class solutions at every step, from the from stabilization, as we move on, from the stabilization to extraction, then amplification with NGS, either a whole genome library or with our targeted panels for oncology and other targets. We then have confirmation with our PCR panels, which will allow you to rapidly confirm anything that you have found with the NGS. We have then integrated that into our data analysis with our clinical workbench and our Ingenuity, our Insights program. You can come by the booth and speak with us about any of these solutions that we offer. Now, though, you're probably most interested in hearing how this is working in the real world, so I will hand it off to Dr. Laurence Lapointe, who is the CEO of Clinical Genomics. Dr. Lapointe has had a lot of success, both scientifically and in building companies that are involved in the field of cancer screening. Right now, he is the co-founder of Clinical Genomics, which is deploying a test for colorectal cancer which was co-developed with CISRO. It's available now in the US, and they have in partnership with Quest Diagnostics. and they're continuing to add and to build on to the testing that they have available. So thank you, Dr. Pointe.
Thanks, Angela. Thank you all for your time today to learn about clinical genomics and our work on circulating tumor DNA to develop a blood test for colorectal cancer. First, a little bit of legalese. Although I'm very appreciative to QIAGEN for giving me the chance to speak to you about our work. Make the emphasis that all of the slides here and the discussion is either mine or from Clinical Genomics, and it does not represent the views of other people, including QIAGEN, Quest, and some of our other partners. So at Clinical Genomics, what really inspires us is the idea of applying innovative healthcare solutions to very clearly defined unmet needs. And perhaps a really good example of our work there is in the colorectal cancer space, where for the last 20-odd years we've been involved in starting with a screening test in the late 90s to introduce a user-friendly colorectal cancer screening kit. This year we'll sell about 3 million of those things in the United States and around the world, to our more recent work to develop a blood test for cancer at the other end of the spectrum for patients that have been previously diagnosed and treated for cancer. And I'll make a couple of shout outs here to QIAGEN and appreciate their time. QIAGEN been a great partner for us through this whole period. So why colorectal cancer, and why do we focus particularly on this disease? For two reasons. First, because it's a high impact disease. This is the #2 cancer killer in many countries in the developed world. But perhaps more importantly, this issue of unmet need. Colorectal cancer has some really well-defined application questions of clinical diagnostics. So here's a disease that if you can't catch it early, and the conventional understanding is the disease takes somewhere between, call it 6 to 10 years to develop from the earliest lesions to late stage disease. If you catch this cancer early, before it invades the bowel wall, before it spreads, you have a 5% chance of dying from that disease and a 95% chance of surviving out past 5 years. Conversely, if you don't detect the disease early and it's picked up only at the metastatic stage, those numbers flip, and you've got a 95% chance of dying within 5 years from colorectal cancer. So this is a pretty interesting diagnostic opportunity where there's some clear benefit. What does the #2 cancer killer look like in terms of the numbers? Well, globally, it's about 1.4 million people are diagnosed each year for colorectal cancer, and about half of those people will succumb to the disease. So given this really clear opportunity of early detection, you have really great outcomes and trouble with later stage detection, the question really has to be asked, why is the mortality rate of colorectal cancer still remain so high? There's really two reasons. And the first is that despite the fact that there's been level 1 medical evidence for the better part of 30 years that shows that you can reduce the mortality and morbidity of colorectal cancer by early diagnostic tests, most of these stool-based tests are not particularly friendly for patients. The compliance levels across the board are relatively low. And in simple terms, poor compliance, not doing tests, results in these cancers being detected either late staged or completely missed until the very late stages, and therefore with devastating effects to the patient. Now, the second reason why the mortality rate of colorectal cancer is so high is a little bit more complex, but really lays the groundwork for the work that we're doing now. So even if a patient follows guidelines, carries out a colorectal cancer screening test, either colonoscopy or any of the tests that are out there, including one of ours, they are diagnosed as positive, they end up having colonoscopy, and colorectal cancer is detected. The patient is they undergo curative surgical resection, effectively cured from colorectal cancer, and these are great outcomes for many of these patients. Across the board of all stages of cancer, there's a 30 to 40% chance of that cancer relapsing. Often when that relapse occurs, it's quick, and this is true of many cancers in terms of cancer relapse. 80% of the time within 2 years, in colorectal cancer, 90% of the time within 3 years. Frequently, that relapse or recurrence is in fact not to the local bowel. It's to distant mets to the liver or the lungs. effectively a stage 4 cancer, regardless of what the initial stage of diagnosis was. And going back a few slides, understanding about, once that disease is out, the mortality rate is extremely high. Colorectal cancer surveillance monitoring is testing in patients that have been previously diagnosed and treated for colon cancer with the intention to identify those patients, those recurrences, as early as possible so that the doctors can intervene either with further chemo, subsequent, further surgery, resections, or what have you. In the United States, there are approximately 1 to 2 million tests will be done this year to ask that question, did the cancer come back? Now, the follow-up here, what this slide shows in this quick pictograph is you've got a cancer diagnosed, patient undergoes surgery, often totally curative, or yes, they suspect curative. Stage 3 patients will undergo chemotherapy, and then they have this 3 to 5 year monitoring process where they're doing an annual CAT scan. These 1 to 2 million blood tests are being run in between those CAT scans to ask the question, does the cancer come back, and should we accelerate this downstream imaging? So, in the US, and in fact many countries around the world, there's only one currently guidelines-approved blood test. It's based on a 1970s technology called carcinoembryonic antigen, or CEA. The good news is that when CEA is positive, and the patient has, in a patient with a recurrence, a colorectal cancer recurrence, the outcomes are often really favorable. So if the test, when it works, it actually works pretty well. The challenge with CEA, and this is a relatively old glycoprotein, is that it has really well understood deficiencies, and in terms of both first sensitivity, it misses a lot of cancers, and I've got a quote here I'll come back to, but in fact also because it has a relatively high non-specific positive rate that really causes a lot of confounding at the oncologist level about what do they do about these positives. It's affected by smoking, non-specific inflammation, various other things. So here's a quote from a Cochrane review at the end of 2015. CEA is insufficiently sensitive to be used alone, but yet here, looking at the bottom, trying to improve sensitivity by adopting a low threshold is a poor strategy because high numbers of false alarms are generated. So clearly an unmet need here with respect to the current only guidelines available blood tests for monitoring. At this conference, there's a lot of discussion of, call it liquid biopsy, free circulating DNA. In particular, this is an interesting thing. I'm not sure how to fix it. I don't speak German. Are you okay? You guys, sorry? No, you're not okay. No? Is that okay? We'll try this. Your slides have gone off the rails here. Sorry. So I think while you do this, I make the point that looking at circulating tumor DNA, we in fact think that there is a utility for it. And although there's lots of work, sorry, thank you very much. Apologize for that. We think so the question needs to be asked, can circulating tumor DNA really inform this question of colorectal cancer recurrence? In particular, and there's a number of flavors and a lot of different things, concepts that people think of when they think of circulating tumor DNA. We're going to speak about one very, very particular version of this that I'll come to in a second. But in the broader spectrum perspective, the circulating tumor DNA we think offers two really important advantages over classic biomarkers like CEA. First, with a relatively short half-life, estimated to be about 2 hours in vivo in circulation, the suggestion is that when you have a positive circulating tumor DNA assay of whatever flavor version of the assay you're measuring, whatever biomarkers, then it tells you something about the real-time specificity. So a positive assay can tell you about a tumor that's currently in the blood. And setting aside some of the work on looking at prognostic tests and risks, we're really focusing on circulating tumor DNA from a tumor in the body, giving you that really important real-time specificity. Secondly, with modern technologies like PCR and other technologies that are looking at really low copy number detection, there's a possibility now to pick these cancers up at really what is there's various terms being used, micrometastasis, but the one I like here is minimal residual disease. So you're picking these cancers up in single copy levels in 4 to 10 mils of plasma, so a lot of plasma, but really detecting these cancers at these earliest events. in the colorectal cancer field, this is in fact something of a challenge, because oftentimes these ctDNA assays are positive even before the disease is imageable by radiological means. So, but looking forward, this idea of detecting cancer at minimal residual disease, I think, really suggests lots of possibilities in terms of chemotherapy and resection at the earliest possible event. So at Clinical Genomics, we've spent about 10 years looking at circulating tumor DNA, or in fact, probably a better way to put this, 10 years looking at biomarkers for colon cancer that we think have application in blood. And along the way, we've looked at lots of different things. We've explored CTCs. Just our base technology was on messenger RNA, gene expression changes up and down. We've explored exosomes, microRNA, CTCs more recently. But where our research led us, in particular, was to focus on epigenetically silenced gene targets. The reason why we're interested in, we think that that's in hindsight been something of a serendipitous approach for us, is that hypermethylated gene loci, in particular for tumor suppressed genes that are turned off, really offer two key advantages. First, at least empirically in the markers that we've wound up in our product, these gene changes, these hypermethylated epigenetic silence events, occur really early in the neoplastic process. In our case, even in the adenomas that precede the actual invasive carcinoma. Then looking downstream, there's a lot of discussion around, particularly at ASCO this year, around the nature of tumor heterogeneity and tumor evolution, and in particular the impact of that on mutation-based testing. So looking at hypermethylated gene loci seems to, in fact, address both of these questions. Early, but also persistent, so you can have an assay that gets you all the way through the process. In our discovery work, starting with a number, so a little less than 400 downregulated gene biomarkers that we were interested in researching, we then carried out research to identify which of these downregulated biomarkers were particularly associated with a hypermethylation event. Obviously, gene expression is affected by lots of transcriptional control elements, but we were particularly interested in hypermethylation events because this gave us something to, in fact, measure in the blood. So about 1 in 6 of our downregulated markers turned out to be hypermethylated targets. And then the top part of this triangle here, looking at starting with 23 methylation markers, really depicts and shows the results of sequentially increasingly large clinical studies that we did. Importantly, in each of these studies that we published and through this research process, for us, none of this was discovery. These were all assays that were really bedded down. We really asking the question in a proper validation sense, what are we looking for? And where we wound up was a series of about 5 biomarkers here shown in this penultimate triangle at the top of this graph of BCAT1, IKZF1, which I'll come back to in a second, but also IR4, GRASP, and COM. COM is perhaps an interesting example of a marker that previous to our work wasn't identified, and COM is CAHM is colorectal adenocarcinoma hypermethylated. So one of the genes that we named in the process. But ultimately, what we identified was for our product, which is we've now described as Colvera, we selected 2 gene markers that are hypermethylated early in the neoplastic process and then persist through late-stage cancer. Branch chain amino acid transaminase 1, BCAT1, and the Icarus zinc finger gene, IKZF1. Now, the stories behind these, our work was really about identifying diagnostic tools, not biological targets, for instance, for drug therapy. But, there's a lot of interesting science on these markers in the last couple of years. Amino acid transaminases, they're involved in branch chain differences being part of cell cycle regulation. And the Icarus zinc finger marker is now identified as involved in the tumor as a tumor suppressor gene that seems to control Notch signaling, which is important in the crypt formation in the colon. So, an interesting post hoc story for us. But focusing on our product here, from here on out, I'll call it Colvera. I'm referring to this 2-gene hypermethylation assay. How does it work? Well, this is the slide here, and I very thankfully put a big QIAsymphony here on the slide. We've worked very closely with QIAGEN over a number of years. So the first step involves extracting DNA from a relatively large volume of plasma. So we're talking, in our assay, it's 3.9 mils, which is sort of where we wound up because that's effectively what we get from one purple top tube of blood, whole blood. So a relatively very large amount of plasma that we're working with, and that was pretty tricky. So for us, developing technologies in partnership with QIAGEN, pardon me, to handle that high volume was really enabling for us to continue our work. Because we're looking at methylation signals, our next step is then to do bisulfite conversion, which is a pretty horrible process for DNA. It's boiling DNA at 96 degrees in ammonium or sodium bisulfite. We then involve another cleanup. All of this is done with QIAGEN solutions, where we wind up ultimately back on a QIA Symphony and an AS module that prepares our PCR plate. We currently run this, we're pretty much PCR agnostic. We're running on an LC480. But importantly, once we run those assays, we're running them out 40 to 50 cycles, any positive signal for us in either of these two gene targets, so any detectable methylation by the two PCRs is positive in our case. We're not looking at bioinformatics. There's no models, logistic regressions, or what have you. These are very simply interpreted. Any detectable methylation in these two gene loci is a positive assay. Treat it for suspicion of recurrent colorectal cancer in our case. As a small company, we really are highly dependent on our partnerships, first with Quest, who has been involved with us from our founding, and more recently over the last couple of years with QIAGEN. And I thought it was important to point out three particular areas that we've enjoyed some success working with QIAGEN. First, they've working both in Australia, where a lot of our primary R&D was done, but also working with a group in Hilden in Germany, and more recently in the US. We've had great access to early chemistries. They've been helpful to work for us. What that meant in terms of our assay was this developing an automated formula extraction platform, so something that's commercially reasonable and viable. They provided great support to instrumentation from the tech support team, so lots of program changes to our various instrumentation from QIAcubes and Symphonies. They've been responsive to help us do that. And now looking forward, our business model is really not about selling kits and making, being an IVD manufacturer, although we have some history in doing that. So the availability working forward of, packaged components allows us to focus on the things that we want to focus on, meaning the clinical conversion of our assay, not, making in-house prep. So QIAGEN's been a great partner for us. Now, in terms of the data, does the test work? Well, the short answer is yes. We've published a number of papers, particularly over the last 24, 36 months. I've got a couple of these ones shown here. I should say that I'm very happy to offline provide the clinical data if someone's looking for a particular paper or understanding something in particular. But starting with our work in tissues way back in 2007-8, it was very clear, again, going back to an earlier point I made, that 99% of the colorectal cancer tissue specimens relative to matched normals in the peri samples that were collected had expression of either BCA-T1 or Icarus or both. So these had very high penetrance, if you can use that term here, in terms of hypermethylation in these tissues. Importantly, and this was the break for us, was that the ability to move from tissues, though, about 66% of cancers across all stages have evidence of BCA-T1 or Icarus methylated in circulation. So that's a very high sensitivity. To put it in perspective, that's not too different than the classic level 1 data that was captured with guaiac-based FOBT for colorectal cancer screening. So these tests are pretty good. And in particular, they have exquisite specificity. A 94% specificity really helps us understand what we're doing as an assay. From a clinical chemistry perspective, the simple version is that having a really specific target, and not all hypermethylation targets are specific, allows us to really amp the assay and have confidence that when we get a signal, it's not a noise event. So over the last 2 years in particular, we've got a series of publications in about 3,700 prospectively enrolled participants, all in a blinded, finished assay format, showing the efficacy of this device, of this test, in patients with colorectal cancer confirmed by colonoscopy. More recently, in October of this year, in fact, only a couple of weeks ago, we are really excited about publishing our work now, taking that background work and understanding how the device works, characterizing it in colorectal cancer, patients with colorectal cancer, to applying that to a recurrence monitoring setting. And this is an excerpt from a paper that published in Cancer Medicine, again, that was October of this year, showing that in prospective blinded specimens in patients that are being monitored for colorectal cancer recurrence and then are diagnosed by CT radiology, Colvera, this 2-gene assay, was positive about twice as often as this classic glycoprotein. So this is a really big difference. This is not an incremental improvement that an oncologist is kind of scratching the head and wants to ask how much the test costs. This makes a really big difference to these patients, or it can anyway. And for us, this work really lays the groundwork for our clinical strategy, which is in several parts here. First, we're very pleased to announce that we are bringing up a CLIA regulated lab in Bridgewater, New Jersey, and that lab is coming online as we speak. We have collaborated with Quest, who has been a co-founder of our company going back to 2006, who has agreed to use their 2,100 patient service centers from around the country to collect plasma and send them to our lab. So this is a really, really big advantage for us, all of which then allows us to run this initially in a lab-developed test format. We can do this because colorectal cancer monitoring, that's acceptable. We instantly are bringing an FDA approval process to bear as well. The lab-developed test initially is being offered as an adjunct to CEA. There's no reason why you would want to replace CEA initially. It's guidelines approved, and they're already collecting the blood. So from our case, it's a pretty simple conversion exercise of just collecting another tube of blood for Colvera. But we're offering it. The intended use is for patients that are using CEA to undergo monitoring for colorectal cancer recurrence. And now, finally, I'd like to make one kind of motherhood point about clinical genomics, and in particular reach out to anyone here that may be interested in speaking after the talk. We have a pretty strong view that this kind of mantra of ours, that advocacy leads to adoption. Our history, first starting 20 years ago in screening and being somewhat successful in inducing FIT to what we hope is going to be the future of Colvera, is really predicated upon our ability to collaborate with people, with key opinion leaders in the area. We believe, bake the science, make the test available, let the truth speak for itself, and ultimately that's how you can achieve success in this space. So if someone's interested in understanding the assay further, or even wants to talk about collaboration, very happy to talk to you. And finally, looking forward, what's next? I guess it is important to point, so we do have a number of projects ongoing with QIAGEN and others to look at how we'll take this thing forward. A very important point that is critical to circulating tumor DNA testing is this issue of patient sample stability. It's pretty, I think it'll be obvious to many of you, but when you're looking for circulating tumor DNA or free circulating DNA, whatever you want to call it, you really don't want to be contaminated by that white blood cell DNA that sometimes is lysed in the specimen handling. So we're really excited about some work to develop improved sample stability kit with QIAGEN to really take away that problem from having lots of relatively unskilled pipetting in the field. So I think this is a big step forward. And then looking beyond that, in colorectal cancer monitoring, it's about 1 million to 2 million tests being run each year. So even with this solution that I presented earlier in terms of the multi-step, this high throughput, robust method, scaling is a real issue here. So as we look to sample and answer, we're quite excited to collaborate further and really look at kind of the next generation of how we're going to take these things forward. In summary, I thank you for your time. We believe that the promise of ctDNA diagnostics to identify and treat cancers at the most deadly stage, metastasis, is really exciting, and we think that, and we're looking forward to presenting further results as we go forward. So thank you very much.
If anybody has questions, we can take them now.
Yep. I'm sorry, I can't quite hear you. No, that's a great question. So no, they often, relatively late stage cancers, there's a global hypermethylations that are going on later on. So you often see simultaneous methylation at high levels very late. But early on, it's really important. It's why we chose 2 genes, and really specific genes so we don't confound specificity. But in our case, we do see at low levels, particularly in the early stage cancers, 1 gene going hypermethylated before the other. And I'll also make a mention that in our assay development phase, intra-hypermethylation was important. So one of the 2 genes, in particular Icarus, has CPG sites that in fact get further methylated within so picking your probes is tricky for your RT-PCR sense, because they in fact, hypermethylation seems to increase as part of a general proposition through the process of etiology of the disease in general. But identifying them early, any methylation of either marker for us in 2 genes gives us the sensitivity we're looking for. I can't hear a word. I'm sorry, son.
Okay.
That's good.
Just regarding the DNA that you get, if it's unusual certificate is not white blood cells,
Yeah.
I think white blood cells is like DNA and certificate too. So how is anything they need? How will you be sure that the DNA that you have is not or is not from that? How can you be sure?
Yeah, look, no, well, empirically, obviously, we can't be absolutely sure what we're looking at. What we want to do is minimize the possibility of that white blood cell DNA contaminating. So it's probably a more interesting question. So I guess I should step back and say, we did select these markers because in big pools of DNA, they're very infrequently methylated in the normal population. So you got to start with a very specific background concept anyway. But unlike, for instance, in a mutation testing where this issue of copy number and contamination can be really a problem, we can amp that assay. Now, is it possible that we have white blood cell DNA in some of these assays? Of course we do. What's important is that we minimize that. And it is true that if you really overwhelm these assays with a lot of DNA, then your chance of having a false positive event for whatever reason becomes a challenge. But for us, it's a quality control step. We use beta-actin as a means of testing whether or not we think the level has gone to a certain level where it's potentially contaminated, and we see that. And often, if there's a lysis event, a poorly treated specimen, or they just can't seem to pipette the right level and they're going to grab some of that buffy coat, then the beta-actin goes off the roof and we have them retest. Great. Thank you very much.